Greetings and salutations, everyone. My name is Dambaba. Arguably, one of the goals of modern neuroscience research is to understand how nervous systems produce behavior. One of our working hypotheses is that nervous systems and neurons use spikes to communicate with each other or to encode information, and I'm here using the term information very loosely. The interesting thing in our study of nervous systems and how they produce behavior is that unless we're recording from inside a cell, we're never actually recording spikes directly. So an important problem in neuroscience, before even getting to the point where we can ask questions about how the brain encodes behavior, is the ability to infer spikes using the recording technologies that we have at our disposal. What I'm going to show you in this talk is how to use autoencoder architectures for the purpose of inferring spikes from electrophysiology data. Similar principles could be applied to the problem inferring spikes from modalities such as calcium imaging. Earlier in the course, you were introduced to autoencoder architectures that I call vanilla autoencoder architectures in the following sense. So the autoencoder architectures that I will show to you rise or come up starting from a probabilistic generative model, a broad probabilistic generative model of signals that are the superpositions of patterns that repeat in time or in space with different amplitudes and locations. Starting from that model and learning algorithms for inferring the parameters of their model, I will show you how to derive these autoencoder architectures for discovering patterns. Electrophysiology is arguably one of the simplest modalities we have at our disposal for probing the activity of the brain and of neurons. What you're seeing here in the green trace is a recording from a single electrode that was inserted in the hippocampus of a rat. The deflections that you're seeing here at the bottom are potentially spiking events. And I'm saying potentially because some of them that have a lower amplitude could or could not be spiking events. The ones that have high amplitude are clearly spiking events. What I want to point to your attention here is the fact that whenever we insert an electrode in the brain, it picks up activity from the neurons that are surrounding the electrode. In this particular case, we have knowledge that there's primarily two neurons surrounding this electrode that it is picking up. Spike sorting is the problem of assigning each of these deflections, when they are actually spiking events, to the neuron to one of the neurons surrounding the electrode that produce these events. It turns out that we can pose this problem as what's called a sparse coding problem. Let us again use this example of these data where there are primarily two neurons active. So we're going to treat the data as Y, and we're going to construct a matrix H that will have two components, H1 and H2, because we're considering primarily two neurons here. And H1 and H2 have the following property. Each neuron, when it spikes, there is a medium from where the electrode here is and the location of the neuron that one can describe as an impulse response, or a spike template. People would typically refer to it as a spike template. The matrix H1, its first column is the spike template of the neuron started at the position 0 here in my time series. The spike template is typically on the order of a few milliseconds. Now, what H1 consists of is a spike template delayed by different amounts and all possible delays that could occur in the signal. Similarly, H2 is a spike template for the second neuron positioned at all possible delays. Now, what we're calling the spike events is represented by this vector x here, which has two components, x1 and x2. And x1 and x2 are both sparse vectors because we're going to assume that the neurons spike sparsely. And the time or the locations where their entries are non-zeros tell us when a given neuron spikes, and their amplitude tell us what the amplitude of that spike was. So linear algebraically, we write this as just y equals hx. We'll go to the mathematical details of this in the upcoming slides. For now, the main point is that starting from this probabilistic generative model, we're going to see how it gives rise to autoencoder architectures that are going to be able to solve what I'm calling the spike sorting problem. So if you see here, we started with the green trace and we produced two traces, a blue one and a yellow one, corresponding to the spiking activity of each of the neuron, as well as the spike templates or the spike shapes corresponding to the neurons. Why is it interesting that one is able to go from this generative model to the autoencoder architecture? Well, for one, starting from this generative model, being able to give rise to an autoencoder architecture is an example of what I call explainable AI. The weights of this architecture, as I will show you, are going to be related to this generative model. Second, because we can deploy these autoencoder architectures in neural networks on GPUs, we're going to be able to solve these sparse coding problems or the spike sorting problem very fast. If you notice, what you're looking at here is just a very generic example of the autoencoder architecture that you were introduced to in this course. And what I will show you is architectures that look like this. And the primary thing that I want you to pay attention to is that the weights of the autoencoder architectures that will develop together are related to the generative model. So you have H here, and you also have H here in the encoder of this autoencoder. And the decoder of this autoencoder also has weights that are related to H. So in some sense, these autoencoder architectures are going to have a lot of structure that is inherited from the generative model. So, when you insert an electrode in the brain, again, what you record is what I call this data trace here, this black trace here, where there are deflections that correspond to spikes, but these deflections correspond to spikes that could come from any of the neurons surrounding the electrode. And what you're seeing here is noise, instrumental noise, or electrophysiological noise. Now, in this particular example, because I generate these data, it turns out that there are three neurons that are surrounding this electrode, which I'm calling a blue, red, and a yellow neuron, each having three spikes and a distinct shape of the spiking event as seen from the electrode's perspective. What is the purpose of spike sorting? The purpose of spike sorting is to take the black trace from the previous slide and produce the location and the amplitude of the events from each of the neuron, as well as the spike template for each of the neurons. So we're going to take the black trace and we're going to assign to every deflection in the black trace, we're going to assign it a color, we're going to assign it an amplitude, and a time when it occurs. And what I will show you is that we can start with a generative model for the black trace, which is going to be Y here. This generative model and an algorithm for inferring the parameters of this model, which are going to be this H matrix that contains the spike templates for each of the three neurons, and this X vector, which for each spike template or for each neuron tells me when it spikes and with what amplitude. And we're going to turn this into an autoencoder architectures that's going to be able to learn these parameters when trained, as well as the time and the location of the spikes from all of the neurons. So the generative model for this, what I'm calling this shift invariant sparse representation, goes as follows in the equation that is in this bottom. So again, this black trace here is my vector Y, and each of its entries is a y little n. So little n is an index for discrete time here. And we're going to assume that each of the neurons, each of the three neurons, so little c here goes from 1 to 3, and each of the three neurons has a characteristic template, hc of n. These are the characteristic templates in blue, red, and yellow. That is convolved with a sparse vector that contains the timing and the amplitudes of the events from that given neuron with additive noise. So this generative model here, it turns out that we can write down in linear algebraic form as follows, as I explained in one of the first slides of this talk. So this convolution here, if we pick H1 which is the template from neuron 1, the blue neuron, and x1, which tells us when that neuron spiked and with what amplitude, I can build a matrix capital H1 now that's going to be that spike template delayed at all possible locations in my signal. I can repeat this for H2 and for H3, and the vectors x1, x2, and x3 are going to contain information again about the timing and the amplitude of events. So in what follows, I'm going to call the spike templates, I'm going to refer to them as this matrix H1, and I'm going to refer as x1, x2, and x3 as codes or sparse codes. Our goal is the following: given only the data y, the black trace, how can we solve for the template for each neuron and the sparse codes for each neuron? It's an unsupervised learning problem. So one way people have tackled this problem from a mathematical perspective is using optimization. So before I proceed, in practice, what we typically do is, because these recordings can be very long, on the orders of hours nowadays, we break them down into small windows that are amenable to processing. And I'm going to call each of these windows y superscript j, where j goes from 1 to some capital J. So this optimization problem was cast to solve this problem of learning the spike templates and the sparse codes for each of the windows. The math itself is not very important. What's important is that this optimization problem is trading off two terms. It's trading off a reconstruction terms that says that I want to be able to use my estimated spike templates and the times and locations of the events to reconstruct my window yj. But at the same time, I want to make sure the second term here is not too large. And the second term is a term that is meant to enforce sparsity of the codes. We have this constraint here, which is to take care of scaling ambiguities, because you could scale h by some alpha and divide the codes by the same alpha and still get the same product. Now this is what people call a non-convex optimization problem because it involves the product of h and x. So the way people tackle this in practice is using a procedure called alternating minimization. So we're going to assume that somebody's kind enough to give us a guess of what these spike templates might look like. We could take this from some prior recordings that we have. And using those guesses, we're going to pretend that's the spike template for the neurons and we're going to solve for the sparse codes. Call that convolutional sparse coding. Having those sparse codes, we're going to take those timings and locations and amplitudes now and update our guess of the h's. We're going to alternate between the two until convergence. So our first step is, given the filters, we solve a convolutional sparse coding problem. This should be familiar to you as something you've seen in the course. This is nothing but what is called the lasso or L1 regularized regression. So what's interesting about this step is that it is embarrassingly parallelizable across all the windows you have in your data, which therefore means that it is amenable to GPU processing, and you should pay attention to what I just said here because this is going to be one of the motivations for using autoencoders and neural networks to solve the sparse coding problem. Now the step when given estimates of the times and locations of events from the neurons, we want to update the spike templates of the neurons. This step is more computationally expensive because it is not parallelizable over all of the J windows. All of the windows must contribute to the update of the filters of these spike templates. Now, it turns out that those two steps, for reasons that I'm not able to go into the mathematical details of in this talk because of time, you can check out references to our publications at the end of this presentation if you want details. It turns out that the convolutional sparse coding step is equivalent to this recurrence here. And very briefly, what's happening here is that the inside of these parentheses does gradient descent on this least squares loss. And it follows it by a nonlinear operation that comes from sparsity here, and it turns out that that nonlinear operation represented by this nonlinearity here, eta, is the ReLU, because we're using L1. Again, there's a beautiful mathematical theory behind this. You can look into the details in our publications. Now, it turns out that this gives rise to what I'm calling a nonlinear encoder, which is this first half here that stops here. So if you look at this, It only involves operations that have H transpose in them, and H and a value as a nonlinearity, and it takes the data as input. Now my decoder is going to be linear because of the form of the dictionary update step. This equation here is the least squares loss with a linear operation on the code. This will give rise to my autoencoder, and I'm going to use the data Y here and a least squares loss to train this via backpropagation. Again, so to put this in the context of the autoencoder architectures that you were introduced to in this course, what's interesting about these is that because of the generative model we started with, whose goal it is to detect recurring patterns in data, we end up with very structured architectures. And the structure comes from the probabilistic model as well as the algorithms we use for inferring the parameters of this probabilistic model. So we use simulated data to test the ability of these autoencoder architectures to learn ground truth templates. So what we did, we simulated 17 minutes of electrical activity from 3 neurons with a sampling rate of 10 kilohertz and an average firing rate per neuron of 30 Hz. So what's important here is that we have control over the spike templates, and we also have control over where we put the spikes and their amplitudes. We generate the data, add noise to it, and feed those data as the input to the autoencoder, and ask it to learn the spike templates as well as the location and amplitude of the spikes. So we did this. So we initialized the spike templates, 3 of them, so that their angle, the sign of their angle with the true spike template, which we know is roughly 45. The sign of the angle is roughly a half, meaning their angle is roughly 45 degrees. And what we did is that we trained the architecture via backpropagation, and as a function of epoch, we tracked the sign of the angle between the ground truth template, which we have control over, and the one that the backprop algorithm estimates as every epoch. So what you want to be able to see is the following. You start at 45 degrees, and as epochs go, you want the angles to get smaller and smaller and smaller, so that the sign gets smaller and smaller and smaller until you get to 0. And this is what you can see in this plot here. Now, interestingly, it turns out that training the autoencoder architecture, which we call CRESA for Constrained Recurrent Sparse Autoencoder, is 5 times faster than using standard techniques from the optimization literature that solve the convolutional sparse coding step and the dictionary update spec using standard optimization packages. And the primary reason for this is that those are not readily deployable on GPUs. Now, another interesting thing is that for spike sorting, which is after you've learned the spike templates, now inferring the times and amplitudes of events, our algorithm or our architecture, because it is amenable to GPU processing, is orders of magnitude faster than competitors that are based on this probabilistic generative model, one of which is known as the continuous basis pursuit algorithm. What these 2 figures here are showing is that as a function of time in training, and as a function of iterations, our autoencoder architecture and the Sparkle algorithm are able to achieve similar levels of reconstruction error of the input data traces that we feed in. So it's not just that our algorithm is faster, it is faster and it can achieve the same level of accuracy as other algorithms. We also see this when we apply the algorithm to real data. So what I'm showing you here is the application of our algorithm to a data set that I introduced at the beginning of the talk. This is what people refer to as the Harris dataset. This is a set of recordings from the hippocampus of rat using tetrodes. I'm only showing you one of the electrodes here, and it is known for this data set that there's primarily 2 neurons that are surrounding the electrode, and for one of the neurons, the interesting thing is that we have what is called an intracellular recording of the neurons. So we can the people that conducted these experiments recorded from inside one of the neurons, one of the 2 neurons that are surrounding the electrodes. What this means is that we have ground truth data about when that neuron actually spikes. And this gives us a way of testing the ability of whatever algorithm one might come up with to either detect spikes or not, or say that certain events are spikes when they're not. And this is what the next plot is going to show you here. This is called the receiver operating characteristic curve. And what it shows you is, given your input data trace, where you do not know which events correspond to which neuron, because now you have ground truth data from one of the neuron, here's what you can do. Come up with any algorithm, apply it to a data trace. It's going to assign certain events as neurons, as spikes, and other time events is not going to assign as spikes. Because we have ground truth data, we can compute the proportion of events that were actually spikes and that our algorithm failed to label as spikes. This is what we call true misses. Now, we can also compute the proportion of events that are false alarms, meaning events that our algorithm said were spikes, but that are not actually spikes from the perspective of the ground truth data. Once you do this, you obtain this curve here, where the best performance you can get is by being at the bottom left corner, 0 spikes missed and 0 false alarms. And what this plot is supposed to show is that our architecture is competitive with continuous basis pursuit, while it is orders of magnitude faster, as I showed you in previous slides. So to conclude, what I did here, what I tried to do in this talk is to show you how to develop autoencoder architectures that are able to detect recurring patterns from electrophysiological activity, starting from a probabilistic model of such signals or such time series, and then developing learning algorithms for the parameters of that probabilistic model that now can be translated into what I call explainable autoencoder architectures. Autoencoder architectures that have structure beyond the vanilla architecture that you were introduced to in this talk. And finally, I showed you that these architectures could be applied to spike sorting, the problem identified as assigning events in a time series of electrophysiological data to the neuron that produced them, as well as being able to estimate the amplitudes of these events. I want to thank you for your attention, and I want to thank the organizers of the NeuroMatch Academy for their hard work, and I want to give a special shout out to Conrad Cording for inviting me and for helping to convert these slides to the NeuroMatch Academy format. Thank you for your attention.